So we are starting with the nucleus, and I added a diagram for them. So by my right now, you can see the object of the you can see the nucleus in its simplest form. So the nucleus is what? A cellular nucleus is what? A circular and central placed organelle in a cell. The nucleus is surrounded by what is called a nuclear envelope. Nuclear envelope means what? Cell membrane of means what? Nuclear membrane. You can either call it the nuclear envelope or the nuclear membrane. Now, looking at this picture, you notice that the membrane does not form a full circle or a complete circle. It kind of like have what breakages, which allows what tiny spaces called pores. So now these pores are called the what nuclear what pore or nucleopores. So these pores are called what nuclear pore. The surrounding of the what nuclear membrane, the circular surrounding of the nucleus is called the what nuclear envelope or nuclear membrane. Now, the nucleus houses the genetic information of the cell. Now, this genetic information, okay, let me continue step by step. Now, the nucleus contains genetic information of the cell, right? Okay. Now, there is something in the nucleus. The arrangement of this information is what? In form of something called chromatin. Chromatin. I know most of us have heard about chromosomes, chromatin. I think chromatid as well. So, what is a chromatin? A chromatin is like the storage, the packing form of what? DNA. DNA in eukaryotic are linear and so elongated. Now, when it comes to the nucleus, so the cell can efficiently pack them very well. They arrange them. Okay, let's do a little imagination here. I know most of us here have what? A needle and a thread. Now, when you unwind your thread, it can easily tangle, right? When you unwind your thread, sometimes if you want to now lose the thread to use it and do something, it can what get tangled and you can spend hours trying to lose it. So the people that produce the thread, for them to make things easier for you, they decided to wrap the thread around something like what? A tube. And when they wrap the thread around the tube, now you have your what? Thread, that's your wool. So if you come to a tailor shop, you can see different colors of thread. You can see red, blue, green, black, and all. So all these threads are what? Rounded up. They are wrapped up in what? They are wrapped up around what? A small tubes. Likewise, so now I can tell you now that let's just say now that human beings they learn this process from what? Nature. Nature has a way of what? Organizing things. Things that are easily tangled, things that are so long, they have a way of what? Packing them and making them very what? Concise. Okay. Now, in the nucleus, the DNA that is very long and linear is wrapped around proteins. These proteins are also called what? Histone proteins. Please write this down. You can easily see it in your object question. DNA are wrapped around what? Histone proteins. There are 8 core histone proteins. So DNA are wrapped around histone protein.
You say it's histoprotein.
Histone, yes. Histone. HISTONES. Histone proteins. Sorry. ONE. Histone proteins.
ONE.
Yeah. Histone protein. So
histone protein.
I'm not hearing you.
Is it histone protein?
Yeah, histone. Histone is what? A type of protein. So you can call it a protein called histone. So DNA plus histone protein form what? A chromatin. Okay. So I was telling us that the chromatin is a packing form of what? The DNA. So the chromatin is basically made up of 2 components. A DNA
You said
that is wrapped around.
the chromatin is what, sir? You said that the chromatin is what, sir?
Okay, just listen. I will explain this now. Let me recap everything I've said so far. Hope you are listening, right? The chromatin,
Yes.
please, off your mic. Is kind of like I'm hearing myself. Is repeating. Okay, so the chromatin is a packing form of the DNA inside the nucleus. I told us earlier that the DNA is linear and very elongated. So for the cell to properly arrange the DNA very well, it wraps it around histone proteins. Now, when it wraps it around
Please
histone protein, I should do what?
Please spell the histone protein.
Okay, so it is spelled H-I-S, H-I-S, then tone, T-O-N-E, histone, H-I-S-T-O-N-E.
Okay.
All right. So the chromatin is made up of what? DNA plus histone proteins. So also inside the nucleus, now look at this image again. You see a dark spot, and then you will see diffused. You see something like thread line there. That thread line you're seeing there is what? Chromatin. Now, this dark spot you're seeing is what is called the nucleolus. This dark spot is what is called the nucleolus. It is what where ribosomal RNA are produced. So inside the nucleolus, you produce what? Nuclear proteins. Inside the nucleolus, you produce what? Nuclear proteins. You also produce what? Ribosomal RNA. Okay. Now, I think in subsequent class I will explain the function of this ribosomal RNA. Okay. So
You
the
said
next thing,
the nucleus. Hello, sir. You said that
I'm
the
listening.
nucleus is where ribosomal RNA is produced.
Yes.
What is the nucleus now from this diagram?
What's the word? Sorry.
The nucleus. Since
Now the diagram.
circle there is nucleolus.
Yes.
Where is the nucleus?
Now, first of all, the nucleus is what? A circular organelle. This whole thing you are seeing now is the nucleus. Let me see if I can save. I think the first slide has a picture of a cell. This cell is in 3D form. Now, can you see this? Can you see this image?
Yes.
Now, this image is your cell. Now, inside this cell, this is just the 3D representation.
Which image?
Are you seeing this image?
Yes.
All right. Now, this image is your cell. Inside, you can see a more circular portion here. This one I'm circling now. Let me see if I can change the color. Now, what I'm circling here is your nucleus. Are you hearing me? Now, this is your nucleus. Inside that nucleus, there's another darker small spot. This smaller one I'm circling now. This is your nucleolus. Now, look at your right. You'll see the label in there. The whole of everything here is your nucleus. Inside is the nucleolus. Is it clear now?
Yes.
All right. So in this diagram now, we zoomed in on the nucleus, that circular organelle. We zoomed it on it. Now, when you expand it, you now see that nucleolus clearer, and you now see the nuclear pores as well. Someone's mic is on. Do you have a question?
Please, can I screenshot?
Yes, you can screenshot. You're free to, okay?
Please, I have my light on. Is it affecting?
No, it's not affecting it. So lastly, the DNA controls genetic information and protein synthesis, and it is found in what? All eukaryotic cells. Okay, are you done to the question in the slide?
Yes.
Okay.
Okay.
So the next organelle we have here is what? The mitochondria. The mitochondria. So the mitochondria is what? A double membrane organelle. Someone's mic is on, please. The mitochondria is what? A double membrane organelle. And like I told us earlier, what? Someone's mic is still on.
So I didn't
Mike.
get the last point
Someone's mic is
you
on.
mentioned in DNA.
DNA or nucleus.
DNA, the second to the last point. The last point said that is found in only eukaryotic cells.
Okay, yes. The nucleus, not DNA.
That
The
is,
nucleus is found in only eukaryotic cells.
huh?
The nucleus is found in eukaryotic cells.
There's a statement you made before this last one.
It controls all what genetic. It carries genetic information and what controls all protein synthesis. Okay, so moving on to the mitochondria. Please, everyone should mute your mic. Just mute your mic, okay? Thank you. So the mitochondria has what? A double membrane. It is a double membrane organelle with the inner membrane fold, with the inner membrane folding into what is called a cristae, and then the outer what? Membrane. Now, the inner membrane folds into structures called what? Cristae, Cristae. Now, the purpose is to increase what? The surface area. Now, the matrix, that's the inner part of that what? Mitochondria, contains enzymes for cellular respiration. Cellular respiration is what? Production of ATP. Okay. Now, it produces ATP through aerobic respiration. That's it is still what responsible for what? Energy production. Now, the major important part I want to take out from here is that the mitochondria is more active. It is what found more in abundant in active cells. Now, I told us that mitochondria is responsible for energy generation, right? So since it produces energy, it means that the number of mitochondria you found in cell depends on the cell's energy need. Okay, I think we left our factory for a very long time now. Let's come back to that our factory now. This factory of ours, let's say we have a very big factory now that we can cover what a whole university school field. Now, in this factory, we are not just producing cars, we are also producing aeroplanes, and let's make this imagination very wild. We're also producing boats and ships. So, do you think the generator used to power someone's room or used to power a mechanic's workshop can now power that factory as big as that? Now, the generator that you can use to power probably your building, that's your room, will not be enough to power a whole building. That same generator cannot run what? A school. Now, you cannot say this generator now can what? Run a factory as big as this. So what I'm trying to infer from here is that the more active, that's the more work a cell does, how active a cell does determine the number of mitochondria it is going to have. Why? Active cells need energy. Okay? So mitochondria is found in more abundance in what? Active cells. Now, what is an example of an active cell? Your muscle cells. So your muscle cells are what? Very, very active. So your muscle cells have what? Numerous what? Mitochondria. Okay? Now, let's move on to the chloroplast. Time is running. I run out of time. My God. So I've told us earlier that the chloroplast is found only in what? Plant cells and what? In algae. So what enables chloroplasts to produce their own food? Why? They contains green pigments called the chlorophyll. So what is photosynthesis? Photosynthesis is what? Just converting light energy into chemical energy. It traps the what? Energy from the sunlight and converts it to what? Chemical energy in the form of what? Glucose. Also, please take note of this. The mitochondria has double nucleus, double membrane. The chloroplast also have what? Double membrane. Okay. Now we are moving into the what? Golgi apparatus. If you remember what I told us earlier, I told us that the Golgi apparatus is also called.
So please let me screenshot the
Have we got that right? Are you done? Okay, so I'll take that as a yes, right? Okay, so the Golgi apparatus. The Golgi apparatus is also called the what? The post office of the cell. Now, it is a series of flattened sacs called cisternae. This mitochondria folds into what? Cristae. This one is what consists of what? Flattened sac. If you look at the picture here, it seems as if it was flat. That's like ribbon-like and compressed. So this flattened sac is called the what? Is cisternae. Now, the Golgi apparatus, just like a post office, I know most of us were not able when post office was still in vogue. Now, what happens in a post office is this. When you bring in your letter, okay, let's say there's a post office in Anambra now. Okay, now many people bring in letter from different, and this letter is to be sent to different parts of the world country. Some letters are going to Enugu, some are going to Anambra, some are going to Abuja, to what Federal Capital Territory. Now, when all these letters come in, it is the what office or the what post office that will arrange these letters. It will now sort them. Okay, every letter going to Enugu, we need them in this table. They'll gather them here. Every letter going to Lagos, they'll bring them here. Now, when they sort all these things, they now do what? Post the letters. Sometimes they can modify them, they can package them, seal them. Okay. Now, when all this thing is done, they now what? Ship the letters to where they are needed. So that's why the Golgi apparatus also mimics a post office, or rather, the post office mimics the Golgi apparatus because this one is in existence before post office came in. So it modifies proteins, it modifies lipid. Modification can be what? Adding some component to it. Some lipid are added protein components and they are now called lipoproteins. Okay. So some protein can be added glucose components and can be called what? Proteoglycans or glycoproteins. All right, so it modifies all these things. You can add other things to it, and now what? Shifts them to where they are needed. So the Golgi apparatus works closely with the endoplasmic reticulum. I want you to underline that, or rather to write that. It works closely with what? Endoplasmic reticulum. Now, let me take us back a little. Remember, the endoplasmic reticulum has two types, rough and smooth. Rough has ribosomes attached to it, which enables it to produce proteins. The smooth one does not have ribosomes, which enables it to produce what? Lipid. So since it is producing proteins and lipid, it is sending them to the Golgi apparatus. The Golgi apparatus is receiving them, arranging them, and what? Distributing them what? Out. So also it is important in secretion and exocytosis. Exo means outside, cytosis means cell. So combine these two words together, it means what? Sending something outside a cell. Okay, lastly, the Golgi apparatus forms secretory vesicles and what? Lysosomes. Later I'll tell you something about lysosome and perisomes.
what is that he sends outside the cells?
Exocytosis. Is that what you mean? I don't get a question. Hello?
I say, you said that
Yes.
he's something outside the cells.
Okay,
And,
what is sent outside the cell?
uh,
Wait, I explained. Okay, let me come again. I say that it is important in secretion and exocytosis. So I explained that
yes,
exocytosis means sending something what outside the cell. So what's your question?
something.
Okay, something like what? Secretory vesicles. Hello,
Hello? I'm listening, sir.
have you gotten it? Have I answered your question?
You said it's vesicle.
Yes. Okay. Now let me come again. Vesicles are like, okay, how will I explain this now? Vesicles are like wrapping some things. Okay, first of all, I told us that it modifies and packages proteins and lipids, right? Okay, so
Mm-hmm.
how does it package proteins and lipids? When it brings the one in it, wraps them up inside vesicles and now sends them out. So that's why it is called secretory vesicles. Also, it produces lysosomes. Now, proteins make up enzymes. Okay, hydrolytic enzymes are enzymes that are found inside the lysosomes. So it can what produce enzymes and what it can produce these proteins and what wrap them up inside the lysosomes and then sends them out. So the form of sending something out from the cell is called exocytosis. So the Golgi apparatus performs two major functions: secretion and exocytosis. Have I cleared it now?
Okay. Yes.
All right. Okay, so the lysosomes now, and then this same what peroxisomes. Let me explain them now in details. Please off your mic. All right. So the lysosomes, like I mentioned earlier, they contain digestive enzymes that would break down macromolecules and worn out organelles. So lysosomes are what they contain digestive enzymes, also called what hydrolytic enzymes. They perform major in function in what cell defense and apoptosis. Now, the peroxisomes also break down things, but what is break down there is what fatty acid, and it's also what detoxifies hydrogen peroxide. I doubt you see hydrogen peroxide in exam. You can see H2O2. You can write this down. This is the molecular formula for what hydrogen peroxide. H2O2. That's hydrogen peroxide. So two hydrogen, two oxygen. You know, water is H2O. But when you add one more oxygen there, it becomes oxygen peroxide. Do you have any question?
Yes.
Okay.
You said that it does what to hydrogen peroxide.
Okay, just listen. Okay, when I'm done, you will ask your question because I was not done with the explanation. I just noticed your mic was on.
Okay.
So let me recap again. Now, lysosomes break down macromolecules and worn out organelles. Please off your mic. So lysosome breaks down macromolecules and worn out organelles. Now, peroxisome breaks down fatty acid, and what detoxifies hydrogen peroxide into water. So during metabolic activities, metabolic processes, which I'll not go into details here, hydrogen peroxide is produced. Now, this hydrogen peroxide is harmful to the body. It is harmful to the cells. Since you're looking at cells here, it is harmful to what cells. So, what does peroxisome does. Peroxisome contains oxidative enzyme, oxidative what enzymes. So it's now what comes and interacts with this hydrogen peroxide to detoxify it. That's to remove it from that harmful state into water. Okay. Which analogy will I use now to explain this? Let's say you want to wash off oil. Now let's say oil, okay, now when oil stains your plate or you finish eating and there's oil in your plate, if you want to wash it off, you now bring detergent and mix with that oil. And then that detergent to allow that oil to what, come out, that's you can easily wash it off. Or you want to wash your clothes, you mix water with detergent and what, scrub it off. Now, likewise, they say that detergent now what detoxifies that oil, it degrades that what oil and makes the plate clean. I don't know if this analogy will just play out very well here, but this is almost the same thing. Okay, I have a better one. Okay, this goes to the girls. Now, when you are cooking and you now notice that your food, maybe let's say your soup is too salty, what do? You add water. Now, let's say you cook the soup and the soup is very concentrated. That's what we call conch soup, right? Now, when it's very concentrated, the ingredients might be concentrated too. So if you want to reduce that concentration, that saltiness, you add more water, right? When you add water now, the water will neutralize that saltiness. Now, I want us to take this information that something can neutralize the other and bring it inside the cell. After some processes, metabolic activities, the cells can produce hydrogen peroxide. Hydrogen peroxides are harmful to the cell, they're oxidative agents. Now, the peroxisome comes and what detoxifies them. I will not go into details on how it does this. It's a complex process. Now, it detoxifies them into what? Water, which is not harmful to the what? Cell. I'll give us the knowledge I want us to take out from these two because there are some questions. I've been going through your past questions. We have videos already
Yes.
on.
I'm not
Your mic
seeing
is
your
on.
slide.
I did not change it, still there. You're not seeing it like
Okay, it has appeared.
all
It
right.
disappeared
Probably
before.
okay, I think it was network. So if you're writing down something, I want you to take note of these things. There are two major things you should note here. Now, lysosome contains hydrolytic enzymes. I'll repeat. Lysosomes contains hydrolytic enzymes. I hope you're writing it down. Peroxisomes contain oxidases, that's oxidative enzymes. Peroxisome contains oxidases. You can put slash oxidative enzymes. Let me come again. Lysosomes contains hydrolysis or hydrolytic enzymes. Peroxisome contains what? Oxidative enzymes. Secondly, lysosomes are formed from Golgi apparatus. Lysosomes are formed from Golgi apparatus. Peroxisomes are formed from the endoplasmic reticulum. Peroxisomes are formed from the endoplasmic reticulum. And the two of them are what? Membrane-bound organelles. They are what? Lined by membrane. Okay, so moving to the next slide. Yeah. Oh, there's something I forgot to tell you as well. Okay, please note somewhere. Ribosomes, I think I mentioned it. Ribosomes, they don't have membrane. They are not bounded by what is membrane. Ribosomes are non-membrane bound organelles. They are not bounded by any membrane.
Ribosomes,
So
not lysosomes.
no.
Sir?
Yes, ribosome, the ones that produce that synthesize proteins. I forgot to mention that.
Okay.
So ribosomes, they don't have membranes. While lysosome and peroxisomes, the two of them have what? Membrane. They are membrane-bound organelles. So moving to the next component of the cell, we have the cytoskeleton. The cytoskeleton. So the cytoskeleton what contains there are what structures that help what that helps in what they provide the what structural framework of the cell. So cytoskeleton, like I mentioned, means what cell what skeleton. That's the simplest form. Cyto means cell, skeleton means what a structural framework. So cytoskeleton is what the structural framework of the what cell. It tells them what in movement. It tells you what cell division. Now, cytoskeleton contains
Sir,
three. You know,
uh, please, can I get the completion of the lecture later? I'm feeling very sleepy. I'm even dozing off my phone.
a commission of what? Sorry, what did you request for?
Like
For the
completion
slide?
of the lecture. Up, yes, the slide. So I can read from there.
All right, but I just feel that you missed some point I will mention along the way because the slide is not always comprehensive. Okay, we are almost done though. Just hang on. I think this one and then cell membrane. I'll dismiss us. Yes, just these two, we are done.
Okay.
Okay, so let me wrap it up. So the cytoskeleton three main components. Number one is a microfilament. Second one is the intermediate filament. And the last one is what? Microtubules. So now then all these things, what it does is what it provides structure and framework to the cell. The microfilament is made up of what? Thin fibers. Microfilament is made up of what? Thin fibers. It tells
It's, it's made up of what, sir?
thin
The
fibers called actin.
thin.
Yes, thin fibers called actin.
Called what?
Actin, look at the slide, you see there. Actin, ACTIN.
Okay.
So it supports the cell shape. Now, the intermediate filament as well helps in providing mechanical strength. I think your mic is still on. Okay, so cytoskeleton, their general function is what? Maintenance of what? The structural framework of the cell. It helps with what? Transport, cell division, cell shape, and what? Mechanical strength. Okay, so moving on. Now, the last component of the cell we want us to look at is what? Cell membrane. The cell membrane. Okay, I think last week I was using a complex diagram to show the cell membrane, but today I had to look for a simpler illustration. Now, this is a schematic presentation of the cell membrane. Circular head is what is called what? The head region. If you look at it very well, it has two heads, that's two yellow circles up and down. And then inside it, it has some things like thread. This is called the what? Hydrophobic tails. I'll still not go into details here. Probably one day we'll talk about cell membrane and then I'll tell us in details. So the cell membrane is made up of what? A phospholipid bilayer. That's two layers of phospholipid. Phospholipid means what? Phosphorus and what? Bind to what? Lipid. So it's made up of what? A bilayer phospholipid or phospholipid bilayer with embedded proteins. If you can look at this protein now, this protein is looking as if it is forming a channel. This is called what? A channel protein. There are two classifications of proteins here. There are peripheral proteins and integral proteins. This one that crosses the entire length is called the what? Integral proteins. This diagram does not have peripheral proteins though. Don't worry, when we talk about the cell membrane, I will teach us this in details. Now, the cell membrane is a selective permeable membrane. Selective in the sense that it allows some things to enter and it prevents others from what coming in. Now, if you look at this protein channel here, this protein channel what allows some materials to what enter in. It's also called what facilitated diffusion. we'll still look into this in details. Okay. So for now, we are looking at general overview. So cell membrane also contains receptors for communication. They also act as what? Signaling what? They contain signaling molecules. Most importantly, one thing you should take notes from this is that the cell membrane helps to maintain homeostasis. What is homeostasis? This is the first thing you will always learn in physiology. Homeostasis is maintenance of a fairly constant internal environment. Homeostasis is the maintenance of what? A fairly constant internal environment. Fairly constant means somehow close to constant. It should always be around this number. First of all, it cannot always be constant. There can always be variations, but the variation should be so little that it should always be what? Almost constant. Now, what you are trying to maintain constant is from primary school, we have been told our what? Temperature. The normal body temperature is 37 degrees Celsius, right? Now, one of the days I went to the hospital to check my vital signs, mine was 36.7 degrees Celsius. Not exactly 37, but yet still constant. I think the next time I went back, I think one of my visits there again, I checked it, mine was still 36.5. Did you see the fluctuation from 0.7 to 0.5? Yet, it is still what? Around that 36, 37, fairly constant. If you should rise above that 36 point to around 40, that's what? We are now running temperature. If you should come down, you now what? Suffer what? You now what? Start having cold. So homeostasis is your body trying to what? Conserve some things at what? Regulated what? That's at what? A specific measurement. I think your blood pH is around what? 7.4. That's all you should know for your cell membrane. Lastly, the cell membrane is also described as what? A fluid mosaic model. Fluid mosaic model. Fluid in the sense that it is not static. It is not like a rigid wall. Your cell membrane is not like your fence in your school. No. It is what? It has this fluid structure. Now, why is it called a mosaic? I don't know if most of us in primary school, there is this thing we did, mosaic. They were kind of like tell you to create a design. You bring broken tiles and everything. Now, when you create that, it is what? Mixing things with different patterns, different colors and all. So looking at this cell membrane, you will see purple, you will see yellow. Don't mind the color. I'm just using color to explain this to you. It does not have all these colors in actual sense. But when you look at it, it does not have a specific structure. Seems as if different components are mixed into it. So it's called what? A fluid mosaic what? Structure. A fluid mosaic what? Model. So I think that's all for cell organelles. Okay, so the next to last slide for this is practical tips for drawing plant cell. These are things you should know by now. Use what? Sharp pencils and of course don't shade. Draw clear line. Don't use that. I really must have about that. Why? Because your body is not what? A perfect straight line. Your cell can never be what? A perfect straight line. So you should what? Draw with your what? Hand. Draw with free hand. Label everything accordingly. If you like, you can, if needed, you can draw according to scale. And then practice need labeling. If you look at this labeling, they are very neat and they are not overcrowded. Also, use round or oval shape to represent cell boundaries. Draw your nucleus central. Your nucleus will be at what? Center. Label every other organelle. So that's just the tips for drawing. So in summary, cells are the basic unit of life.